is called a pyranose, os sugar. A puran is essentially a 6-carbon ring, or 6-carbon sugar with an oxygen inside of it. Notice not all 6 of the carbons are in the ring. There's a methyl group here coming off of it. Okay. And what I kind of mentioned before the break, depending on where the hydroxyl group is on what's called the anomeric carbon. So this carbon here, carbon number 1, the carbon that had the carbonyl in it when it was an open chain, if we have this in an axial position down, and again, we're always going to orient the ring in this kind of dimensional space with the oxygen in the upper right-hand corner. If it's down, then we call it alpha. So both of these came from a D glucose, right? So with the hydroxyl group on this side. Then when the ring closed, depending on where that carbonyl was positionally, it'll be down or up. Or it can be considered being trans to this group here, this methanol group, or cis to that 6th carbon, okay, that methanol group. So you can think of it as up or down, cis or trans, but that gives us these 2 distinguishing features. Okay.
And that's what makes it like alpha or beta then, or?
Yes.
Okay.
If it's down, it's alpha.
Okay.
Or down or trans, it's alpha.
Okay.
If it's pointing up or cis, it's beta.
Okay.
And so lactose is broken down into galactose, and so beta galactose is going to have that hydroxyl group pointing up, and beta galactosidase is actually an indicator that's used in molecular research because it turns, so colonies, bacterial colonies, can have a gene inserted into them that when induced, it will express beta galactosidase, which turns blue. And so if you want to know if you have colonies that had that gene replaced with what you want to do, so in recombinant DNA, you take a bacterial plasmid, it's a circular genome of a bacteria, and you can use enzymes to cut it open and put in whatever gene you want. So one of the first applications of this, this is great. Speaking of sugar, diabetes, so insulin. They used to get insulin from pigs. Literally, they would just harvest a bunch of pigs' pancreas. And that, especially now, wouldn't cover the amount of people, the demand for insulin. So one of the first recombinant technologies to ever come out was to take a bacterial plasmid, this circular genome of E. coli, and then you use these enzymes to cut out a certain region, right? So then that piece falls out. And you say, okay, let's put the gene for insulin in there. And then now you put this plasmid back into the E. coli cell, and you induce that to start making that gene, and then you purify out the insulin. So the insulin that's now used for patients with diabetes comes from bacteria, GMOs. Evil GMOs, right? GMOs are great if it's going to save your life, right? If it's GMO in your bread, then oh my gosh, terrible. I'd be facetious if you didn't. I don't have an issue with GMOs personally, but So, now I know. So what you can do is you can take a bacterial plasmid and you can say, all right, I want to put the gene in there that encodes beta-galactosidase, okay, which is a sugar. And when I put it on this petri dish, this agar, it will, if it's made, if we make that sugar, it will turn blue. So you look at a plate and you say, okay, that is what I'm going to use as a starting material, and it's going to have what are called restriction enzymes. These things that sites on either side that we can use enzymes to cut that out, and then put whatever gene we want in place of that galactosidase. So then, when we transform that into E. coli cells and we plate them, we'll get some colonies that are blue and some that are not blue. They'll be kind of whitish. So we'll make the black ones not blue. And then you can say, oh, okay, well, that one didn't work. I didn't replace the gene there because it's still making the beta-galactosidase. Here, that worked. No blue pigment. I'm going to pick that colony. I'm going to grow it up in a big vat. I'm going to induce it to make whatever gene I put in there, say insulin, and I'm going to purify it. So it's it's just an indicator to see if we've done our job right.
So you put that as an indicator, and then you'd put like another gene that you actually want on there, and then if the indicator shows up, you know
that you didn't replace it, you didn't swap it out.
Yeah.
Right? Yeah. Because, again, it'll make... So I'm actually going to, for my micro class this fall, there's a kit that you can buy to do CRISPR. You familiar with CRISPR?
Oh, the editing?
Yeah, the Cas-based genome editing. It's actually a bacterial antiviral mechanism. So bacteria evolve this defense that when a virus, called a bacteriophage, enters and then inserts it, well, it doesn't enter, it just inserts its DNA into the cell. It can recognize it and chop it up. And so they've used this to now be able to edit certain parts of the genome. And so you can do it, you can use CRISPR and then use beta-galactosidase to see if it actually worked. If you modify the region that has beta-galactosidase, then it won't turn blue. But the beta, the reason it has beta in it is because that hydroxyl group is pointing up in that group. And so again, this is how we represent it, okay? We put this in the upper right-hand corner, and then the anomeric carbon is going to be just to the right, okay? And then we can do this chair structure, right? We can bend it into the chair, but you got to make sure that you keep this in the upper right-hand corner, okay? If we did this, the oxygen's in the upper right-hand corner, but it's not in the rear. Oh, I see. Based on this, it's coming in the front, right? And then here it's kind of in the middle. So the only reason it's important to have this orientation is so that when we start saying things like beta and alpha, you know which carbon you're talking about, because these are going to have multiple hydroxyl groups on them. And so the reactions. Turns out that sugar is soluble in water, right? We all know that. Juice, right? Why is it soluble in water, do you think?
It's a polar molecule. A lot of oxygen.
Yeah, because it has all these hydroxyl groups on it. So it's going to readily dissolve in water, and that makes it hard to purify out using organic solvents, our conventional OChem methods of purifying it. And so what chemists have done is actually converted sugars into ethers or esters so that they can then use their organic solvents to pull it out of whatever solution they're wanting to, right? Because if you're trying to get sugars that are in a polar environment separated from the other polar molecules, it's going to be challenging to do, right? And you can't use a non-polar solvent. So the trick around that is to just use these reactions to convert this glucose into, in this case, we have an ester, right? So this acetyl group is just depicting this right here. So we have an ester here, or an ether here. And not only do we do it to the anomeric hydroxyl group, we do it to all the hydroxyl groups. So we convert all the hydroxyl groups into either esters, or we can turn them into ethers. Then we can purify it out. Okay? So that's one of the more common reactions using sugars, other than just, of course, breaking down disaccharides and polysaccharides into monosaccharides or vice versa. That's going to be hydration and dehydration reactions. The other thing that's important is talking about the formation of these rings. All right, we talked a little bit about that. How do we close that ring up? Does it make a hemiacetal if it's an aldehyde, or an acetal if it's a ketone? And when we do that, when we go, for instance, from a hemiacetal to an acetal, which also is what happens when you put two sugars together. So when we form a disaccharide from two monosaccharides, we end up taking the hemiacetal, this hydroxyl group here, and linking it to another sugar molecule. So this is a very generic, representation. But for a sugar, this R group would be the other sugar ring that we're attaching to. Therefore, getting rid of the hemiacetal and making the acetal. When we do that, it's called a glycoside, or a glycosidic bond, glyco sugar. So glycosides are disaccharides and polysaccharides. Or we could just straight up modify a monosaccharide. So here we have glucopyranose, again, 6 carbons with an oxygen in the ring. It's beta because this hydroxyl group on the anomeric carbon is pointing up. And in the presence of methanol and an acid catalyst, we end up getting essentially an ether, right? So we went from a hemiacetal to an acetal. And you'll get different abundance depending on where that methanol comes in and attacks. Okay. If it attacks up here at the top versus if it comes in at the bottom. Okay. And so here's a mechanism where we take our monosaccharide and we essentially turn this into a good leaving group, right? Turn this into a water molecule. It pops off, and then we have this resonance stabilized carbocation. So the lone pair from that oxygen in that ring come down here and form a pi bond, and it just jumps back and forth here until we can then attach the alcohol to it, or another sugar, which sugars are alcohols, right? Because they have OH groups on them. So the alcohol could be something like this, right? Where we're just making an ether, or the alcohol could be another sugar molecule. in this case, instead of doing another sugar molecule, we're bringing in methanol. And if it comes up here from the top, then we end up with beta. If it attacks down here at the bottom, we then get alpha. It's on the bottom or it's trans.
So it's not like preferential one way, it's just kind of how it happens to attack?
It's somewhat preferential. 66% is going to be alpha to 33% beta. And I'm guessing that it's because, well, I don't know. I would think there's more spinach down here, so I would think logic would say that maybe it would be higher. I don't know why it's 2/3 of the time is going to be here, or 1/3 is up top. Not quite sure. It might be that the lone pair on that oxygen in the ring, it's avoiding, maybe it's avoiding the lone pair here.
Oh, okay.
Coming down here.
It's right
Even
next.
though this hydroxyl group is here.
Because that one's axial or radial on the other.
This is axial,
Uh-huh.
and in this case, it's like equatorial. Yeah. It might be, just that the hydrogen is going to tend to be out here rather than, it might be that this hydrogen tends to not be next to this hydroxyl group. It tends to be out here more, so it's just the access is easier. Those are my guesses, though. So take it for what it's worth. All right. How much time we got? Reducing sugars. A reducing sugar means that it can be oxidized. Right? So what happens is that we can take the ring, we can open the ring up, because it does open, right? If you looked at that equilibrium, the arrow wasn't pointing one way, it was just mainly in the hemiacetal direction. But rings do open, and when they do, if they are an aldehyde, they can be oxidized, okay, into essentially carboxylic acid. Okay, so we go from glucose to glucaric acid or aldaric acid, and that is a reducing sugar. If it's a ketone, not going to happen. Okay, and so there are tests to see if you have reducing sugars or not. You can use Benedict's solution. Have you done that? Anybody done that before? No? Okay. It's blue because it has copper ions in it. And when you put the copper ions in something that can reduce it, well, then it goes from C2 plus to C1 plus. And then with that comes a color change. It goes from blue, copper ions are blue, copper 2 plus ions are blue, copper 1 plus is green, and then non-ionized copper is kind of a reddish, brownish, color. Right? So when you have copper outside, if you've seen copper guttering and stuff, it's not so much prevalent anymore because people steal it all the time. But it has a green patina to it.
Yeah.
Right? That's copper 1. Copper 2 plus is usually going to be in solution. So you can see if things are reducing sugars, because if they are reducing sugars, they will reduce the copper from 2 plus to either 1 plus, which means it's got some reducing sugars and some non-reducing sugars. And then if it's all reducing sugar, it'll change it to like a reddish hue, because it goes to just straight non-ionized copper. Okay? So, how can we predict whether or not a sugar is reducing? Well, we have to look at it and say, for one, is there a hemiacetal? Because what is a hemiacetal indicative of? An aldehyde. If it's a ketone, it's going to be an acetal. So we need to know if there is a hemiacetal or an acetal. And so in this case, look at the anomeric carbon, and which one do you see here? You see ace or hemi?
Acetal.
Yeah, right? Is it an OH group or is it an OR group essentially? This is an OR group. So that means it must come from a ketose. Ketoses can't reduce oxidizing agents. They can if you use a really strong oxidizing agent, then you can make it happen. But using something like Benedict's solution, it will not happen. Okay. So we would say this is a non-reducing sugar. What about A? Here's the oxygen in the ring, so this is the one coming down is the anomeric carbon. Is this hemi or is it acetal?
Acetal.
Acetal, because it's an OR group, so non-reducing. Here's the oxygen. Here's the anomeric carbon. Hemi, so it is a reducing sugar. And then C, here's the oxygen. Go down to the right, anomeric carbon, what's coming off? Hydroxyl group. Hemiacetal must be a ketose. Therefore, it is a reducing sugar. And so here are some examples of sugars in nature. And pretty much all of these are reducing. So glucose, it's reducing. I tried to get my intro to OChem students to pee in cups to see if they had sugar in their pee. And then we could, if there is, we could test it for reducing sugars. And none of them did. So I told them they had to taste their pee. And I doubt any of them did that. I hope none of them did that. But glucose, right? Oxygen, anomeric carbon. So it's reducing. Oxygen, anomeric carbon. Here, it's reducing. Galactose, that's a byproduct of lactose. Anomeric carbon, reducing. All monosaccharides are going to be reducing sugars, okay? When we get to disaccharides, it depends on which carbons are involved. So maltose is a reducing sugar, okay? This anomeric carbon is not, we would say that that's not reducing because OR, right? But this one, anomeric carbon, O. So that ring can open and close, right? Depending on equilibrium. And so it could reduce. And so this is a sugar that probably, if you put Benedict's solution in there, it'd probably turn that intermediate green. It wouldn't go glucose, fructose, galactose, all would be red. Because they completely reduce the carbon from C2 plus to C. There's Cu2 plus to Cu. This, only part of it is doing that, so you might get a green. I would guess you can get a green. This, however, sucrose is non-reducing. Sucrose is a disaccharide of glucose and fructose, so it's made up of reducing sugars, but this is the anomeric carbon, and it is an OR. And it turns out that unlike maltose, that maltose binds not to the anomeric carbon. This is the anomeric carbon. But with fructose, it's binding to the anomeric carbon. Here, it's right here, but we flipped the molecule, so you gotta kind of flip that in your mind.
Yeah.
But because the anomeric carbon is an OR and this one is an OR, there is no hemiacetal to reduce. Okay. Questions?
So the reducing, it's the sugar itself that can be reduced, right? It's not a. So
No, the sugar's getting
It's doing
oxidized. It's doing the reducing. So redox reactions. So reducing is a gain in electrons. Oxidizing is a loss of electrons. When you put it in the presence of an oxidizer, so for instance in this case, if it reduces it, it turns it into Cu plus, right? So it must have gained electrons, and that's going to be green. If it reduces it to Cu, that's red. If it doesn't reduce it at all, it stays blue. So how does something reduce something? This is where it gets this is where I always confuse my Gen Chem students. If you get oxidized, then you are reducing something, right? If you are oxidized, then you are the reducing agent. So the sugar is getting oxidized, and in doing so, it reduces whatever is in there to oxidize it. So that's why it's called a reducing sugar.
Okay, so the copper in this case is the oxidizer that is
Oxidizing
oxidizing
the sugar.
the sugar and reducing itself. Okay.
Yeah. Disaccharides, di-2, right? Put two monos together, you get di. So maltose, as we mentioned, it's reducing. It's called 1,4-alpha glycoside because it has a linkage between the anomeric carbon, which is 1, and not an anomeric carbon. This is the anomeric carbon, right? 1, 2, 3, 4. And it's alpha because the anomeric hydroxyl group is pointing down, or trans to the methanol group. Okay. Here, cellobiose, also 1,4. Sucrose, though, is going to be 1,1 because it's involving both of those anomeric carbons from the glucose and the fructose. All right, cellobiose is what breaks down into cellulose, which we can't digest. Right? Okay. Now, it turns out that alpha and beta disaccharides have at equilibrium can be both representatives. Why? Because again, that relationship of the ring and the open chain, sometimes it does this, okay?
It opens
So
up.
we can have a sugar and have this ring open up. And when it opens up, this hydroxyl group can flip up and then it closes again.
Okay.
Okay, so it can go back and forth, opening, flipping, closing, opening, flipping, closing. So you get a gamish of these two anomers, if you will. They're anomers because the anomeric carbons have both alpha and beta representatives. going back to the very beginning of lecture, Lex, you said lactose, it's a disaccharide, right? Sure is. Galactose, glucose. Beta pyranose, beta pyranose. So they're both beta. This is coming up, and the alcohol or the hydroxyl group here was up. So that's why we have this formation. And that's why you have to have specific enzymes. An enzyme that, this would be beta as well, but if we had an alpha, here's an alpha, right? That linkage, that hydroxyl group is pointing down. And so it becomes even more prevalent when we get polysaccharides because it's going to change the whole shape of that macromolecule. And so enzymes, right, it's lock and key. It's all based on shape. So having something here and something here is going to be a different shape than having them more parallel to each other as if we had two betas. And so you need a specific enzyme to break this down. You need lactase. And people who have insufficient levels, that means they don't make enough endogenous lactase, or they don't make it at all, are going to be intolerant. And so the easy workaround is to actually take, I think it's called Lactaid, or there's different versions, but it's essentially lactase. It's the enzyme. You take it first, you eat it, now you have it in your digestive system, and then when you consume dairy, that exogenous lactase will break it down for you. Okay. From there, lactose can go and change into beta-galactosidase, and that's this blue color that's expressed or induced in these E. coli cells. Okay. Lactose can also be used by bacteria to form lactic acid. Does it say that? Yeah, it says that. To form lactic acid. That's through fermentation. So if anaerobic bacteria, meaning there's no oxygen, are utilizing sugars in terms of lactose, they can break it down into lactic acid as a fermentation byproduct. Okay. So lactose is going to have these D conformers. All right. Okay. And then sucrose. See how sucrose has a different shape? It's alpha and beta. And an enzyme for that is going to have a different shape than an enzyme for lactose. And look, it's 1,2 glycoside. 1, 2. I thought it was the anomeric carbon. I guess that is the anomeric. Well, so it's coming off of here, so they must be saying that that's 1.
That's weird.
I'll have to look into that. I would think that that's the anomeric. I guess it could still be anomeric because this could be up or down. Maybe that's why, but it's not 1. It's they number it 2 because they start with that one. Yeah. Usually though in rings you don't start counting outside the ring. So let me check in on that. All right, how are we doing? Oh, we're past time. Sorry. This is just kind of overview, high-level stuff, though. Polysaccharides are multiple monosaccharides linked together, cellulose being one of those, right? You can break it down into cellobiose, but this is kind of a long chain. Amylose, we can digest amylose. In fact, it begins in our mouth with the first enzyme ever discovered, amylase, salivary amylase. It has a slightly different conformation than cellulose. You look here. then amylopectin. Pectin is sugars in fruits. Pectin is also used to make jams and stuff. If you want to give it kind of a gelatinous structure, you add pectin. It's just a sugar. And then amino sugars are just going to be sugars with amine groups added to them. All right. And now we get into relevant things like DNA. So DNA is ribose or deoxyribo. For DNA, RNA is ribonucleic acid, DNA is deoxy, and we'll talk about that when we get into nucleic acid. So I'm rushing through this stuff because this is really just kind of a primer for nucleic acids. But suffice it to say, when we have a sugar here, this is a ribose, and we put a nucleotide on it, right? Then now we have an amino sugar.
Okay.
And ribonucleoside, this is going to be RNA. The ribo means that it has this hydroxyl group. DNA, deoxy means it's lost in there.
Okay.
We'll talk about that. We'll talk about that I guess in a couple weeks. Next